especially for this, but you don't get to see me. I'm pretty humbled to be able to introduce Katy to come and talk at this symposium. Katy is currently endowed chair in cancer research at MD Anderson, professor of stem cell transplantation, and multiple other leadership roles at Anderson. Katy started out a PhD work in London, in University College London and Imperial College London, and then seems worked at pretty much all of the big famous teaching hospitals in London, Imperial College, Harefield, the Royal Marsden, and quite a stint at the Hammersmith Hospital, all of which we used to get transplantation donors from. She did her another postdoc at NIH in around 2004, and moved to MD Anderson in 2012. So it's quite an honour to be given somebody's CV. I'm familiar with Katy's work that's relevant to me and my work, but when you get to do something like this, you see all this enormity of somebody's achievements, she's listed as a consultant in 25 companies. She's won over 25 awards, like prestigious awards, including American Society of Hematology most recently. There's multiple patents that Katy is the leader of, and 285 publications. So amongst those publications are the work in the NK cell field. So Katy's work has completely revolutionized the role of NK cells in cancer therapy and essentially brought them to the forefront of cellular therapies worldwide, really. It's my great pleasure to hear her describe some of this work to you today.
Thank you very much, Paul, for this very kind, I'm not sure I'm deserving of this introduction, but thank you so much. And really is an honor to be presenting today. I'm very sorry I can't be there in person, but hopefully sometime soon. But what I'd like to focus on is obviously on our NK cell work. But before I start, these are my disclosures. For the purpose of this presentation, I'll be presenting some of our work around CAR19 NK cells. I was licensed to Takeda. Some of our work around NK cells and bispecific engagers, that was licensed to Affimed, and I'll briefly mention some of our work around TCR engineered NK cells, which was licensed, which resulted in a new code as listed here. So I don't think I need to really explain to this audience what a chimeric antigen receptor is, but very briefly, a chimeric antigen receptor brings together the antigen recognition site of a monoclonal antibody with a signaling endodomain of a T cell or an NK cell, so that when the immune cells engineered to express this chimeric antigen receptor, it will recognize the tumor like an antibody would, but would signal and kill like that of a T cell or an NK cell. As I'm sure you're all aware, since 2017, multiple different CAR T products have received FDA approval, all for hematologic malignancies, mostly B-cell malignancies and multiple myeloma, with response rates ranging anywhere from 40 to 90%, depending on the indication. But of note is that all of these CAR T products are autologous, which then means that the patient will have to have their own T cells collected, sent to a specialized lab for manufacturing, and then infused back into the patient. So it's a single treatment, which then means that the product is extremely expensive. And many patients, whilst waiting for their product to be manufactured, have progression of their disease or don't even have access to many of these therapies. So for this reason, a number of groups, including ours, asked the question of, well, what if we use an allogeneic cell donor? So that from one healthy donor, we can manufacture multiple doses that we can then potentially biobank these, so that when a patient comes, they can just have the cells thawed and infused at bedside, thereby increasing speed of treatment, reducing costs, and increasing accessibility. Now, there are certain challenges, of course, to using an allogeneic donor for CAR engineering. For starters, no one really knows what's the best source of allogeneic cell therapy. We know that with certain allogeneic cells, such as allogeneic T cells, there's a risk of graft versus host disease unless the cells are engineered to delete the endogenous TCR. There's always the concern of limited persistence of the allogeneic donors. This could be intrinsic to the cells. So, for instance, natural killer cells normally have a lifespan of 10 to 14 days in the absence of cytokine support. And of course, allogeneic cells are subject to mismatch by the recipient T cells and NK cells. As I mentioned, the choice of cell type is still not very clear. Different groups are looking at engineering alpha beta T cells or gamma delta T cells versus NK cells, iNK T cells, macrophages, cytokine induced killer cells, you name it, they're all being engineered and tested. And what's very important to note, and I'm going to come back to that, not all healthy donors are created equal. So just because you're going after and picking immune cells from an allogeneic donor doesn't mean that you will see the same effect, the same response with all donors equally. So we asked what would we require from an ideal donor? Well, ideally, you don't want to have any requirement for HLA matching. If you really want to have a off-the-shelf source of treatment. But also, you don't want your donor cells to cause graft versus host disease. We'd want to have defined selection criteria so that we know how to pick the healthy donor. We want the cells to persist, at least perhaps not for the lifespan of the patient, but for longer than a week or 2 weeks. Ideally, we'd want our cells to have multiple mechanisms of killing so that the recognition is not just reliant on the CAR recognizing its cognate antigen, thereby reducing the probability of relapse through CAR target antigen loss. We'd want the cells to be safe. And then we also want to be able to have a scale up of manufacturing, because after all, we would want to try and manufacture hundreds, if not thousands of doses from one patient. And we'd want to have a way of freezing these cells so they're post-thaw, then maintain their potency. So we believe NK cells fulfill many of these requirements. And just to give you a little bit of background about natural killer cells, they're part of our innate immune system. They have certain characteristics that distinguishes them from that of T cells. And in the context of cell therapy, some of these characteristics are related to the fact that allogeneic NK cells, even if totally mismatched with the recipient, will not cause graft versus host disease. And this is now being proven in thousands of patients where allogeneic NK cells have been infused with no induction of graft versus host disease. And NK cells can recognize their targets through a complex array of receptors that is germline encoded and is expressed on their surface. And in fact, NK cells are they're part of our innate immune system, but also the first line of defense when it comes to recognition of virally infected cells or cells that become abnormal and turn cancerous. So how do NK cells recognize their targets? So NK cells on their surface have activating receptors, inhibitory receptors, cytokine receptors. But if we focus on the activating and inhibitory receptors, as the name suggests, activating receptors give a positive signal for NK cells to kill. Inhibitory receptors give a negative signal for NK cells not to kill. Inhibitory receptors all recognize either HLA class 1 or HLA class 1-like molecules. And as you know, all normal cells express HLA class 1 on their surface. Activating receptors, at least those for those receptors that the ligands have been described, recognize stress molecules or DNA stress molecules that are upregulated on the surface of abnormal cells. So what happens in the normal setting is that an NK cell, when it comes into contact with a normal cell that expresses HLA class 1 on its surface, will get a signal through its inhibitory receptor not to kill. So this is a negative signal. And the strength of the signal from an inhibitory receptor always trumps that of the activating receptor. So then the NK cell will then realize that this is a normal cell and therefore will not kill this particular target. What often happens with cancer cells or virally infected cells is that they downregulate HLA class 1 expression on their surface in order to escape CD8 T cell recognition. So now there's no longer an HLA class 1 being expressed. The inhibitory NK receptor doesn't have an HLA class 1 or class 1-like molecule to engage with. There is no longer the inhibitory signal. Now the activating receptor will recognize the activating NK ligand, which, as I mentioned, in many cases, these are stress molecules, and then will mediate killing of the cancer cell. Of course, this is an oversimplification, but this is just basically generally how NK cells can distinguish between normal cells and abnormal cells. So one could therefore, based on what I said, argue that NK cells have certain advantages for CAR therapy in that in the allogeneic setting, they don't cause GvHD, so they could potentially be off the shelf, and one could manufacture multiple products from one donor, thereby lowering cost of therapy. Killing of a CAR NK cell could be mediated both by the introduced chimeric antigen receptor, but also by these activating receptors that I mentioned, their own innate receptors. And as I will also show you, and others have shown, NK cells seem to have a very attractive safety profile in that they don't cause cytokine release syndrome, they don't cause neurotoxicity. And different groups are looking at different allogeneic NK cell sources. Some groups are manufacturing NK cells from peripheral blood by sorting the NK cells, growing them and engineering them. Others are using NK cell lines derived from patients with NK lymphoma, such as the NK92 cell line that's already being tested in the clinic. We are interested in umbilical cord blood, and that's what I'm going to be focusing on, but also induced pluripotent stem cells were taken to the clinic by Fate Therapeutics and others. And also hematopoietic stem cells are being used as a source of NK cell manufacturing. So as I mentioned, we use umbilical cord blood as a source of our natural killer cells for a number of reasons. First is that we're fortunate enough to have a cord bank that was established by my friend and colleague, Dr. Spall, when she moved to MD Anderson back in 2005, and our bank has collected over 100,000 cord units with close to 35,000 units stored and ready for use. And what is important to note is that umbilical cord blood, as well as being a rich source of stem cells, is also a rich source of natural killer cells. We've shown that actually from one umbilical cord blood unit, we can manufacture over 100 doses of CAR NK cells. Now, a couple of years ago, we published our work on a first-in-human trial where we took a banked umbilical cord blood, we thought the cells, we isolated NK cells, and we expanded them using feeder cells, which we call UAPC. And these are feeder cells that are based on K562, the K562 cell line, engineered to express membrane-bound IL-21, 4-1BB ligand, and CD48. and IL-2. And the cells were then transduced with a retroviral vector that was kindly provided to us by Pietro Dotti when he was at Baylor College of Medicine. And this CAR encoded for the CD19 CAR with signaling through CD28 and CD3 zeta. If you recall, I mentioned mature NK cells only have a lifespan of 10 to 14 days in the absence of cytokine support. So this viral vector also encoded for human interleukin-15, and IL-15 is a very important cytokine to support NK cell proliferation and persistence. And also it included a safety switch based on inducible caspase-9, so that if the cells started growing uncontrollably, we could induce apoptosis of the engineered cells by adding a small molecule dimerizer called rimidacid. So we then published our work. This was a dose escalation study starting with a dose of 100,000 CAR NK per kg, going up to a dose of 10 million CAR NK per kg. Patients received only one infusion of CAR NK cells. And these were patients with either a non-Hodgkin lymphoma or chronic lymphocytic leukemia. We reported complete remissions in 7 of 11 patients treated on the phase 1 study. There were no cases of cytokine release syndrome, no neurotoxicity. And despite the fact that the NK cells from the cord blood were mismatched with the recipient, we didn't have HLA mismatch with the recipient. We didn't observe any cases of graft versus host disease. And this is a before and after PET scan of a patient with Richter's transformation. And what was also notable is despite the fact that these CAR NK cells were mismatched with the recipient, we could detect persistence of these cells by qPCR for 12 months or longer following infusion, indicating that a degree of tolerance or microchimerism has been established for these cells to persist. And again, remember our cells express interleukin-15, the interleukin-15 transgene. So this is a follow-up on the original study in 37 patients. Now, as I mentioned, our first trial reported the data on the first 11 patients. And this is on 37 patients with longer follow-up, and where we looked at the overall survival and the progression-free survival of these patients with lymphoid malignancies. And here you can see that somewhere in the region of a third of our patients maintain their remission for a year or longer after receipt of that one infusion of CAR NK cells. Of these patients, only 2 patients had a consolidation with transplantation. The rest of them did not. And these data are therefore not that dissimilar to what we see in terms of long-term remissions with autologous CAR T cells. But then we wanted to see, what is it that makes these 1/3 of patients maintain their remission? Why is it that the rest of them progress? And we looked at many characteristics, papers currently under revision, and there were no patient characteristics that predicted for that. The underlying diagnosis, age, gender, and the age, etc. None of them predicted. Karnofsky score was a predictive biomarker. But the most important element was the quality of the cord that we had used for the manufacturing. So in this trial, each patient received CAR NK cells from a different umbilical cord blood unit, and the cells were infused fresh. And what we found was that patients who had received NK cells that were manufactured from cords that were frozen within 24 hours of collection, and with a nucleated red blood cell count of less than 8 × 10⁷, resulted in a progression-free survival of close to 70% compared to those patients that received cords with time to freezing of greater than 24 hours and an NRBC of greater than 8 × 10⁷. And around half of our patients received cords with this characteristic. And this was just serendipity. At the time, we didn't really know. We were just basically picking the cords randomly. So then we were kind of trying to understand why are these characteristics so important in terms of the quality of the NK cells. Now we know from cord blood from cord blood data that a high NRBC is often an indicator of fetal hypoxia or fetal stress. And of course, if you leave your cells sitting around for a long period of time, they lose some of their potency, they become stressed, etc. So then we went and isolated NK cells from those cords that we defined as optimal versus suboptimal, and we performed RNA sequencing from these NK cells directly ex vivo selected from cords, and we found that actually there was a big difference in their transcriptomic profile. We also looked at their epigenome by doing ATAC sequencing, and again you can see that NK cells from cords with optimal cord were predicted to have high expression of transcription factors important for NK effector functions such as T-bet and EMS, whereas those from suboptimal cords had a transcriptomic profile that was more akin to that what was described with exhausted CAR T cells, such as expression of Fas-Jun. And then in 4 different mouse models, I've just included an example here. We took NK cells from optimal versus suboptimal cords, and we engineered them to express the CAR exactly at the same time, and then we infused them into mice engrafted with tumors. So this was a Raji mouse model. The NK cells from either suboptimal or optimal cords were engineered to express CAR19, IL-15, and you can see that NK cells from the optimal cord could control the tumor in mice much better than those from suboptimal cords. And based on these data now for all of our clinical studies, we are only using NK cells from optimal cords for our manufacturing. Obviously, we're very interested in understanding mechanisms of resistance. Why is it that some patients, even if you give them NK cells from optimal cords, will go on to progress? And so we looked at the CAR T literature, and in the CAR T literature, as you know, CAR T cell exhaustion, antigen loss, the effect of the tumor microenvironment, all had been shown to contribute to resistance and relapse. But also one interesting phenomenon that was described by Michel Sadelain's group is trogocytosis. And this happens when the CAR T cells come into contact with tumor cells. Some of the target antigen is transferred onto the surface of the CAR T cell. And now the CAR T cell expresses the cognate antigen on its surface, and the CAR T cell itself can be subject to fratricide. So basically what happens through trogocytosis is that there is a loss in the tumor antigen density, but also there's induced self-recognition and fratricide. And so Ethan Lee, a very talented graduate student in my lab, wondered whether the same thing is happening with CAR NK cells, and in vivo he followed the fate of NK cells in mice engrafted with Raji tumor, which is a lymphoma model, and he showed that actually over time these CAR NK cells start acquiring CD19 expression on their surface, and this was associated with loss of CD19 expression on the Raji cells compared to controls. And also this led to exhaustion of the cells and to induce self-recognition and actually apoptosis and fratricide. To overcome that, Ethan engineered a strategy where we transduced our NK cells to express also an inhibitory CAR directed against an NK antigen, so in this case CS1, such that when the NK cells came into contact with the cancer cell, so in this case we use the model of ovarian cancer expressing ROR1 on the surface, they could kill the target, but when they came into contact with NK cells that through trogocytosis had acquired ROR1 expression, the inhibitory CAR would bind to CS1 on the surface of NK cells and give us negative signal to the NK cells not to kill. And in an ovarian cancer model, we confirmed that this dual CAR strategy resulted in much better control of the tumor. We also isolated NK cells from the blood of our patients that received the CAR19-IL-15 NK cells, and we subject them to single-cell sequencing. And very interestingly, we found that NK cells in those patients that can control the tumor, so for instance, patient 6 here, have a very different transcriptomic profile compared to those patients that fail to control the tumor with evidence of actually metabolic exhaustion in those patients that go on to progress. And we're obviously putting a lot of effort into better understanding these mechanisms. Then we wondered whether we can take our approach of NK cell immunotherapy to beyond CD19 positive malignancies. So here, rather than engineering the NK cells to express a CAR, we wondered, well, NK cells express on their surface a very important activating receptor called CD16, and this is the most significant mediator of antibody-dependent cellular cytotoxicity. So we wondered, what if we take our NK cells and we basically complex them with a bispecific engager that binds CD16 on the surface of NK cells and a tumor antigen. So in this case, CD30, and the bispecific engager that we worked with was called AFM13, which basically binds CD16 and CD30. And to further increase the potency of our NK cells, we pre-activated them with inflammatory cytokines such as IL-12, 15, 18, before we expanded them to induce them with a memory phenotype. Now, this is very interesting because traditionally, cells of the innate immune system are believed not to have memory. Memory is a feature of cells of the adaptive immune system, such as T cells or B cells. But now there's increasing evidence that under certain conditions, NK cells can also acquire memory-like features. So for instance, when they're exposed to certain viruses, such as cytomegalovirus, but also when they're exposed to inflammatory cytokines, often released by dendritic cells, such as IL-12, 15, and 18. So Lucilla Kirby in my lab took cord blood NK cells, and basically she either expanded them with our feeder cells, as we normally do, or she pre-activated them with IL-12, 15, 18 prior to expansion. Then she subjected them to RNA sequencing, and she showed that these pre-activated and expanded cells, which we call P+C, had upregulation of many pathways related to JAK-STAT signaling, interferon gamma response, and inflammatory response. And then she showed that if she takes these pre-activated and expanded NK cells, and then she pre-complexes them with this bispecific engager, and then she infuses them into mice that expresses CD30 positive lymphoma, these NK cells can mediate CAR-like control of the tumor when compared to mice that received either NK cells alone or the engager alone. We then decided to take that to the clinic, but then we thought that we could also take this opportunity and prospectively validate our definition of the optimal cord selection, which, as you recall, is cords that were frozen within 24 hours of collection and with a lower nucleated red blood cell count. And so my good friend and colleague, Dr. Nieto, led this clinical trial where patients with CD30 positive malignancies, so patients with Hodgkin lymphoma and T-cell lymphomas that had that were either refractory or intolerant to brentuximab, received treatment with these AFM13 loaded NK cells following lymphodepleting chemotherapy. And because the AFM13 ultimately, the bispecific engager will ultimately fall off the surface of NK cells, after infusing the AFM13 CAR-like NK cells, we also gave additional weekly infusion of the bispecific engager alone. This was a dose escalation study starting with a dose of 1 million per kg, going up, then dose level 2 was 10 million per kg, and dose level 3 was 100 million per kg. The trial allowed for the patients to receive a second dose of these, a second cycle of this whole treatment if they failed to respond or if they had a partial response. So we've treated 41 patients to date. And you can see all these patients had very poor risk disease with a median of prior lines of treatment of 7. The majority of all patients had failed brentuximab, also checkpoint inhibitors, and more than half of them had had previous exposure, previous transplant, either autologous or allogeneic, and 4 of them had previous treatment with CD30 CAR T cells. All patients had progressive disease at the time of enrollment. The data were pretty amazing in that we showed that in all patients, our response rate was 92.5% with a CR rate of 65%. And at the recommended phase 2 dose of 10 million NK cells per kg, which was 34 of our patients, our overall response rate was close to 95% with 70% CR. This is a waterfall plot, and this is a PET scan of the patient before and after. Again, there were no cases of cytokine release syndrome, no neurotoxicity, no graft versus host disease. And again, these cells were infused fresh, and it was 1 dose per, we generated 1 NK cell product per cord blood unit. When we looked at the persistence and the duration of the response, however, this was different to our CAR19-IL-15 NK approach in that we put patients into remission, but eventually many of these patients went on to progress unless the patients received consolidation with a transplant, as highlighted here in the stars. So then we wondered whether we could consolidate the treatment further by giving additional cycles of treatment. So we went to FDA and we amended our protocol so that we could give 4 cycles of our AFM13 NK cells. The data are shown here. Obviously, the follow-up is very short, but Dr. Nieto will show a longer follow-up of this at his oral presentation at ASH. But looking at the persistence of the cells, both for the 2 cycles and the 4 cycles, as you can see, this is very different to what we see with our CAR19-IL-15 NK trial, in that, as expected, the cells are basically persisting for a couple of weeks, and then after that, they disappear. Again, because there was no engineering. So then we went back to our CAR NK platform, and then we thought, well, what if we engineer the cells to target a pan-cancer antigen? And then we started thinking about what is the best antigen to go after. And we thought that CD70 is a very attractive antigen because it's expressed on many hematologic malignancies as well as solid tumors. Its expression on normal tissue is limited. It's only expressed on a subset of activated T cells and dendritic cells and also on regulatory T cells. Otherwise, it's not expressed on non-lymphoid normal tissue. CD70 is a ligand for the CD27 receptor. CD70 as a target antigen was already validated in multiple clinical trials with an anti-CD70 antibody called cusatuzumab. So we knew that it was safe, and that we also found that CD70 was expressed on AML blast. So for these reasons, we decided on investing in developing CAR NK cells targeting CD70. So here, rather than using an scFv-based CAR, we decided to use the natural CD27 receptor as a way of targeting CD70, and then we designed 34 different CAR constructs using linking the human CD27 receptor to different transmembrane domains, different signaling endodomains, and different co-stimulatory domains. Remember, our CAR19-IL-15 included CD28, but here we also included other co-stimulatory molecules that are more relevant to NK cell biology. So in the interest of time, I'll skip the in vitro data, but we picked the most promising constructs based on the in vitro studies, and we put them into our in vivo models. And then we also made a CD70 CAR using the crucituzumab SCFV. And in 2 models, one of Raji and one of AML, we showed that construct number 23 resulted in the best control of the tumor. And then we translated this to the clinic, and we received FDA approval for 2 different clinical trials, 2 basket studies, one in hematologic malignancies and the other one in solid tumors. Here, though, what we ended up doing was we took, again, an optimal chord, as I mentioned to you, using our optimal chord criteria. And in the meantime, we also developed a strategy to freeze NK cells. So NK cells are notoriously difficult to freeze. So we ended up spending 4 years trying to optimize the freezing strategy. We tested close to 100 different freezing protocols, and we came up with a way of freezing the cells that gave us similar potency to that of fresh. So from 2 umbilical cord blood units, we manufactured 250 patient doses that would then allow us to cover both clinical trials. So really reducing the cost of therapy. Our transduction efficiency was around 60%. And here, again, this was a dose escalation study, again, infusing the cells after standard flu-cy lymphodepletion, and the cells are thawed and bedside, thawed and infused at bedside. Now, what's important to note is FDA were pretty tough with us, and they said that our first dose level should be a flat dose of 8 million cells. Remember, with our previous, like with our AFM13 NK trial, we went to as high as 100 million NK cells per kilogram. This is just a flat dose of 8 million cells. Just to put it in context, when we do our mouse studies, we give a dose of 10 million cells to the mice. So a dose level 3 is complete, and I would like to share with you the data with for one patient who, this was a patient at dose level 1, who received a flat dose of 8 million NK cells with, as I mentioned, a transduction efficiency of 60%. This was a 24-year-old male with Hodgkin lymphoma, progressed after 8 different lines of treatment, including 2 transplants, one of which was a haplotransplant. You can see prior to infusion, he had very extensive disease, both above and below the diaphragm. And at day 30 and day 60 post-infusion, you can see how the patient's disease is pretty much gone. The patient had no toxicities, no CRS, neurotoxicity, or tumor lysis. And you can also see how his LDH levels within 2 weeks of receiving the cells went into the normal range. So now we've completed dose level 3, and we've observed responses in 7 of 9 patients and in 6 of 8 patients with Hodgkin lymphoma. So we're very excited about this trial, and it's ongoing. We are also putting a lot of effort into developing strategies to target solid tumors. Another attractive pan-cancer antigen is TROP2. TROP2 is expressed in many epithelial cancers, including ovarian and pancreatic cancer. And again, it's a validated target because sacituzumab, a TROP2 targeting antibody, is already FDA approved for triple-negative breast cancer and bladder cancer. Again, using a similar approach as for CD70 CAR NK cells, we constructed different CARs targeting TROP2. We validated their activity in vivo in our mouse models, and now we've received FDA approval for again a frozen and off-the-shelf TROP2 CAR NK cells for ovarian cancer and pancreatic cancer. And here we'll be giving the cells IP. And we've just received the go-ahead from FDA, and hopefully in the next couple of weeks, we will enroll the first patient on the trial. We've again similarly received approval of the TROP2 CAR NK cells for patients with breast cancer and solid tumor. Again, just to mention that from 1 umbilical cord blood unit, we have manufactured 135 patient doses, which will cover both studies. Again, really reducing the cost of engineering. Of course, with solid tumors, the problem is the immunosuppressive tumor microenvironment. And so we were kind of very cognizant of that. And we started a collaboration with our neurosurgeons in terms of looking at how we can target glioblastoma. Here, the challenges are, in addition to the immunosuppressive tumor microenvironment, is that many of these patients are also receiving corticosteroids. And corticosteroids are, of course, lymphocytotoxic. So there's a concern, a problem of iatrogenic immunosuppression as well. So what we did, we published this data a couple of years ago when we looked at glioblastoma surgical specimen. We could find infiltration by tumor by NK cells, but these tumor infiltrating NK cells were very abnormal. They didn't express the cytotoxic molecules or the activating receptors that NK cells should, and they also had a signature of TGF-beta exposure. When we isolated these tumor infiltrating NK cells from the surgical specimen, in addition, we saw that they are incapable of killing targets. And we also showed that they have activation of the canonical pathway of TGF-beta, which is a SMAD2 and 3 pathway. So we thought that, well, perhaps we can use this therapeutically, in that using CRISPR gene editing, we can make our NK cells resistant to the effect of TGF-beta by knocking out and deleting the TGF-beta receptor 2 gene in NK cells. This was work led by Myra Shani and Rafet Bashar in the group. And you can see in these orthotopic models of human glioblastoma cancer stem cells, mice that receive wild-type NK cells cannot control the tumor, whereas those that receive the TGF-beta receptor 2 knockout NK cells can control the tumor nicely. And then we also went and used the same approach to target the glucocorticoid receptor gene called NR3C1. And then we developed an approach to dual knockout both NR3C1 and TGF-beta receptor 2 gene. In our NK cells, we achieved excellent knockout efficiency. And then we put these again in this orthotopic mouse model. We could show that these dual knockout NK cells could very clearly control the tumor. Taking this to the clinic was truly a challenge, because we had to do a lot of IND enabling studies, really following the guidelines, the draft guidance from FDA, in terms of confirming that our CRISPR-edited cells don't have evidence of autonomous growth. There's no evidence of cytogenetic abnormalities, looking at both on-target and off-target editing, confirming that there is no evidence of off-target editing, measuring residual guide RNA, residual Cas9, and doing very extensive animal studies. And just to clarify, we used the ribonucleotide protein electroporation method. So we did all the scale-up experiments in the GMP facility and confirmed that we can do multiplex CRISPR gene editing in about 40 billion NK cells. And we achieved a double knockout efficiency of greater than 80%. Again, we've made a bank of these cells. We've received FDA approval to start a clinical trial, and this will be a dose escalation study where patients will be receiving these NK cells injected intratumorally through an Ommaya reservoir. So the first part will be dose escalation, and the second part will be a window of opportunity arm. The first patient has been infused with no toxicities to date. And finally, in the last couple of minutes, just to talk a little bit about why we put any effort into engineering NK cells to express TCRs. Well, as you know, TCRs recognize a greater number of tumor intracellular tumor associated and tumor specific antigens. Actually, the majority of tumor antigens are these intracellular antigens, as opposed to the extracellular ones that your CARs would recognize. And so we thought, well, it'd be very attractive to use NK cells for TCR engineering, because NK cells naturally don't express TCR, so there's no risk of TCR mispairing. As I mentioned, they don't express GVHD, they don't cause GVHD, so you can use an allogeneic cell source. 40 to 90% of human tumors, especially solid tumors, downregulate MHC class 1 in order to escape T-cell recognition. So they could thereby also escape TCR recognition. So then NK cells have the added benefit of having their endogenous receptors still there even after we engineer them. And then there were already publications out there by others showing that NK cells can be successfully transduced to express a TCR. So then we use this knowledge, and basically using our platform, we went and engineered our NK cells. Just to add, NK cells don't naturally express the CD3 complex. So we basically developed a strategy to engineer NK cells to express the full CD3 signaling complex. So NK cells normally express CD3 zeta, but they don't express CD3 epsilon, gamma, or delta. So we engineered them to express the full signaling complex, but also interleukin 15. In the absence of IL-15, really CAR NK cells are not very effective. And then we co-transduced them with NY-ESO TCR as a model and as a model target that's been validated clinically. And in our mouse models, we showed that NY-ESO TCR NK cells worked as well as NY-ESO TCR T cells. And then again, we've taken this to the clinic. We've received FDA approval now for 2 clinical trials, one in multiple myeloma, a second one in synovial sarcoma and myxoid liposarcoma. Again, here from one umbilical cord blood, we manufactured multiple products that are frozen and available for off-the-shelf use. And again, this will be a dose escalation study where the cells will be thawed and infused. So then if I can just wrap up, I hope I've shown to you really the potentials that NK cells have for engineering. They are very powerful and incredibly safe cells. We're still trying to understand why they're so much safer than T cells in terms of CRS, neurotoxicity, etc. But we can engineer them to express CARs or TCRs. They can be engineered to express more than one antigen. We've done that. In the interest of time, I didn't include the data. We can, as I mentioned, NK cells on their surface express CD16, so they can be combined with antibodies or bispecific engagers. CAR NK cells or TCR NK cells can similarly be combined with these bispecific engagers or antibodies in order to do multi-targeting of antigens. I've shown you how we can engineer NK cells to express cytokines such as interleukin 15 and how this is safe. We can pre-activate them with inflammatory cytokines. We can combine them with CRISPR gene editing to make them resistant to the immunosuppressive tumor microenvironment. They can be combined with checkpoint inhibitors, with immunomodulatory drugs such as lenalidomide, with radiation. And ultimately, I think for the difficult to treat diseases such as AML or solid tumors, a combinatorial approach is the way forward. So I have so many people to thank. It really takes a village, a country, to do this kind of work, and I'm indebted to my incredible team, the scientists in the lab that develop all the preclinical models and all the various preclinical data that then lead to translation to the clinic, the cell engineering group in the GMP that do all of the manufacturing for our clinical studies, my host collaborators in the Department of Stem Cell Transplant, especially E.J. Spahn, my collaborators in the other departments that we work with, our cord blood bank, our GMP facility, and our bioinformatics team. Also our funding agencies, especially the MD Anderson Moonshot program, and most importantly, our patients and their families, and for putting their trust in us and for taking part in our studies. Thank you very much for your attention. Happy to answer any questions.
Before we get to questions, that's a very impressive body of work. Thank you very much for presenting that. Before we get to questions, it's my pleasure to help add another line to your CV by saying that we're going to give you the Cancer Warrior Award, and this is given in recognition for your outstanding contributions and achievements in cancer research and your devotion to training the next generation of cancer warriors. A plaque will accompany this award. So thank you very much. And hopefully a round of applause from everybody.
Thank you so much.
So if you want to ask a question, you can either type it into the chat or put your hand up, and hopefully Lexi will unmute you so you can talk. Not done this one before, so I hope it works. Is anybody able to overcome the Zoom barrier?
If I might ask the first question.
Absolutely. Please do.
Katy, great to see you. Hatem Sbawi, how are you?
Hi.
Wonderful presentation and really exciting work. So I have a couple of questions on the generation of the products from the cord blood. The first point, maybe you've addressed that you're actually able to produce very large number of doses from the same cord. Do you ever see a need for using more than one cord for transplants as done for other products? The second question is related to some of the single-cell studies that you've done. Is there an NK progenitor effect in that you might identify from the single-cell studies in responders versus non-responders? I'm sure you're aware of Steve Rosenberg's data and the data from Emory. Is there an analogy to the NK field for this?
Yeah, great questions, Hatem. Let me answer your second question first. So we are looking to see whether there is an NK signature of responder versus non-responder. We haven't found a NK signature of kind of a progenitor or more immature NK cells and responders versus non-responders. What we are finding is that on single cell analysis, the responders have more of a effector signature with expression of TBET, EONES, granulysin, so many of the genes that you would anticipate. The suboptimal cords are very interesting because they have a signature of stress, a signature of hypoxia, and a signature of exhaustion. So that to us is very surprising and interesting as to why NK cells from a baby that obviously hasn't been exposed to anything should have that kind of signature. And we are now trying to do more kind of mechanistic studies to understand what is driving that, and whether these NK cells are truly exhausted, or is this something that is happening because they're being sat around for too long before they're frozen, or is it the effect of the nucleated red cells competing for oxygen in the bag, or is this something that's happened peri-pregnancy or peri-delivery in the baby? What we do find is very convincingly that actually if we pick optimal cords and use those for manufacturing, we're very likely to induce responses in our patients, which is what we saw in our AFM13 NK trial. The fact that we saw a 95% response rate in patients with really progressive disease, to me was a true surprise. So from one cord, we can make a finite number of doses. We cannot make an infinite number of doses, which is why we are making CAR NK cells from one cord for each trial. But also I like to be able to also validate that you don't want to get to a situation where you're treating all your patients from one umbilical cord blood, everybody responds, and then a second set of patients from another umbilical cord blood doesn't respond. So for instance, our CD70 CAR NK study, where I showed you 7 of 9 patients in the dose escalation have responded, we made sure that we were taking CAR NK cells from both donors that we've manufactured to really validate that we are seeing responses with both donors. Ultimately, for a truly off-the-shelf, if you want to have a donor that is used for all of your trials, you have to work with iPSCs. With cord blood, you still will have to use different donors.
Thank you.
Do we have any other questions? So while we're waiting, I was wondering, you mentioned that the HLA or the mismatched donors, do you find you have to pay attention to the NK cell receptor and HLA mismatch? Does that have any effect on the success?
Yeah, great question. You know, we've looked at HLA mismatch to look at whether HLA mismatch between the donor and recipient has an effect. Our phase 1 trial with CAR19L15, the first 9 patients received cords that were matched at 4 out of 6 HLA molecules, and after that, we just no longer paid any attention to HLA matching. We didn't see any impact of HLA mismatch on the persistence of the NK cells, on the probability of response. For our AFM13 NK trial, the donors and the recipients were not HLA mismatched at all. I think the average number of matches was 0 or 1. And then we saw responses, as I mentioned, in like more than 90% of patients. So I do not believe that there is any need to match the donor and the recipient when it comes to NK cell therapies.
Okay. I have another question as well. Your TCR, is this, I guess restricted is the wrong word, is this restricted to HLA class 1 or do you?
Yes, yeah, no, that's actually a good point. Sadly, we cannot change what the TCR is directed against. So the TCR will still have to recognize an epitope in the context of the of the HLA molecule. So, for instance, with that NMISO TCR study, all patients will have to be HLA 0201 positive. The cord can have any HLA matching or mismatching with the recipient, but the recipient will have to express HLA 0201 for the TCR to be able to recognize the target.
Do we have any other questions in the last 3 minutes? Okay. Well, just to say thank you again for taking the time and presenting your work to us. Hopefully, anybody that wasn't already converted for NK cell therapy being the next frontier should be some way there now. And thanks to everybody for coming. It's a good turnout. So, thank you very much.
Thank you very much for the invitation. You already have a big NK expert, Mike Veneris, here. So I think, you know, again, I'm sorry I couldn't be there in person, but again, thank you for your excellent questions. Thank you for the award. I'm really honored, and look forward to seeing you all again soon.